And thank you, thank you for having me. This is always a great pleasure to join you in this lovely spot and with such an eager group of students as well. Today I am going to be taking off from a lecture I gave a few months ago. Some chairs of microbiology devoted to education in warm spots were gathered, I think, in St. Kitts. And they asked me to bring them up to speed on the HIV/AIDS agenda 2015. So I devoted the first part of that lecture on better treatments, better immune reconstitution, and of course the cure agenda as we currently have it. And you heard a little bit of that yesterday by Matthias Peterlin. Today what I want to do is focus on effective prevention and ultimately bringing the pandemic to an end. All right. There we go. So this slide is just a simple calculus of what it's going to take. Bill Gates captured it many, many years ago, and he said, we cannot treat our way out of this epidemic. So basically what those curves are mathematical modeling of what it would take ultimately to bring the epidemic to an end. And the only line that continues to go down toward that achievement is the one that combines various modalities for preventing HIV/AIDS, in addition to the wonders and wonderful things that have happened with treatment. And I'm going to focus particularly on preventing transmission to women, and not just women, but the high-risk populations, particularly in sub-Saharan Africa, that actually are one of the driving forces in the epidemic. It's what we call epidemic hotspots. This is just an example in rural South Africa near Durban. And what you see there is, if you look at the age group in rural South Africa, the extraordinary incidence in the red-colored area. It's just shown again on the next slide. This is a township, Vilanlila, that's been studied for many years now by Carisha and Salim Karim. And what you see there is in the prevalence in young pregnant women in rural South Africa, beginning at less than age 16. But by the time a woman is 23 to 24 years, and these are quite recent figures, nearly close to 50% of the women have been infected with HIV. So clearly these are high-risk populations associated with genital tract inflammation, associated with acute infections where the probability of transmission is so much higher with very high loads in the semen. So this is really our target group that we're trying to treat. So this is very appropriate for this pathogenesis meeting because for many, many years now, actually most of my career, I've been interested in using studies of pathogenesis, in this case of transmission in the female genital tract, to derive, in this case, principles for vaccine design. So what we're going to be talking about are the very early stages of infection, the transmission of virus across the mucosal epithelial barrier in women, the formation of a small infected founder population and expansion, and the antibodies that are associated with a live attenuated virus infection, SIV delta F, and we'll talk more about that in just a minute, that seem to be the singular correlate of protection in this model. Now, this is a monkey tail, so to speak, because the critical events in pathogenesis and mucosal transmission take place very quickly. We named the lentiviruses for slow infections many, many years ago, actually in 1972, based on the prototypic virus in sheep, Visnavirus, Visnamati in sheep. And in fact, they're slow infections in the way they play out in terms of the pathology and the accumulation of tissue damage. But it's really a misnomer because the first stages of a lentivirus infection actually proceed very, very quickly. So if you look at pathogenesis the way the doyen of pathogenesis looked at it many, many years ago with mousepox, and you break it into the first hours and days, and you break it out in terms of anatomy, the virus is going to cross that barrier. You're going to see that it propagates locally. There's a local expansion. It goes to the draining lymph node. There's secondary amplification there, and subsequently seeding through the bloodstream of the lymphoid tissue reservoir, which is the site where most virus is produced, where it persists in latently infected cells, where it's stored in enormous deposits on the follicular dendritic cell network, and where the pathology, both CD4 depletion and really the fibrotic changes occur over the long course. So those are things that happen out here at about 2 to 3 weeks at the very earliest, and they've already obviously transpired by the time you would see a woman in the early stages of HIV infection. So that's a black box, and what the rhesus macaque, SIV rhesus macaque model does for us, it allows us to look into that black box and see what's going on inside. And what's going on inside are some very fast events in terms of what happens. Let's go this way. And the main thing that's been discovered is actually some perils for the virus at that mucosal surface. The infected founder populations that are first established are quite small. I think you'll hear some exciting new data from Tom Hope, I believe it's this afternoon, about the exchange and the rapid infection and events that take place within the first 48 hours. But basically, there's a kind of a competition that goes on there. And the first founder populations that are actually established as the beachhead at the portal of entry are small infected founder populations. And what that means, as we'll see in a slide or two, is that the immune system has its maximal opportunities to control and prevent at that early stage. Now, it turns out that there also has to be probably a local expansion. So there's distal seeding from that small founder population, actually probably from the inoculum within the first 24 to 48 to 72 hours. So virus reaches distal sites. There's no question about it, as far away as the axillary lymph node and probably even now we think the central nervous system. But it doesn't really take off. It doesn't catch fire there, so to speak, and establish what we mean in infectious disease by an infection. We mean a self-propagating infection in which one infected cell must transmit to at least one other infected cell, or create one other infected cell. That local expansion just increases the probability. You can think of it almost like wave theory, where you have an attenuation of signal as the farther and farther away you get. But obviously, as the intensity of that wave increases at the local source, you have a greater and greater probability of that virus reaching the lymph nodes, both the draining lymph nodes and the distal sites, and establishing infection. So what we're going to talk about today are some principles of vaccine design that have been derived from these studies of the pathogenesis of in the SIV rhesus macaque model of HIV transmission to women. And the first principle for vaccine design is timing and location. So I've told you that in the earliest stages at the portal of entry, most defenses would be operating at favorable odds against a small expanding infected founder population. And it's that principle that I want to examine in a little more detail now. And specifically, the second principle of local concentration dependence. So what we're going to see with the Delta Neph vaccine is that it creates a system, a local system actually, where antibodies are produced. And then it turns out that the female reproductive tract, immunologically speaking, has evolved an incredibly beautiful design, where antibodies, IgG antibodies, are not only made at that site, but they're actually concentrated by the neonatal Fc receptor, and we'll talk about that in a little more detail in a minute. But what that means is it brings very high, intense concentrations of reactants, the antibodies, with the virus on the path of virus entry. And we think that's one of the mechanisms by which this vaccine works. So just to remind us all about Delta Neph, in a landmark paper nearly 2 decades ago in Science, Ron DeRosier's lab described this variant virus in which the Neph gene was knocked out. Virus turned out it grew really quite well, grew perfectly fine in tissue culture. But when animals were infected IV with this Delta Neph virus and subsequently challenged with wild-type virus, they were protected. And really quite extraordinarily high levels, very robust protection. And in fact, this is just a summary actually from many years ago by Wayne Koff on the live attenuated viruses as the most effective means, the most robust protection against all routes of challenge, basically. Now, unfortunately, not long after Ron's group described that, Ruth Ruprecht discovered that first in neonatal monkeys and then in adult monkeys that eventually SIV Delta Neph would cause monkey AIDS, basically. So there were clearly safety issues. To some extent, I think, certainly said that we were not going to move forward with a live attenuated vaccine concept anytime soon. But the whole idea of a vaccine that really worked well suggested that if you could identify the correlates of protection with that vaccine, it might provide principles to enable the design of an effective HIV vaccine. And that's what we've been working on now for close to a decade. Now, the protection I'm going to be talking about today is really a maturation of protection. It's a very peculiar phenomenon that was described early on. And that phenomenon is after you inoculate this virus, the attenuated virus IV, you get a robust immune response against that virus that actually peaks around 5 weeks or so. If you challenge the animals at that point, there's no significant protection. But if you wait until 15 or 20 weeks, there's very significant protection against very high dose challenges. And just to delineate what those challenges are, we challenge these animals twice in the same day, separated by 4 hours, intravaginally and atraumatically, with 10 to the 9th viral particles and 10 to the 50 TCID50. We do that for 2 reasons. One is it consistently infects the naive animals. And secondly, it also creates events, a probability of seeing things in the tissues when you actually examine the tissues, what I call directly observed pathogenesis. Because what you want to see when you look in the tissues, what are the events that are taking place? What are the interactions between the virus, the infected cells, and the immune response to it? So that's the model. We'll be talking about challenging animals at 20 weeks when they're protected. And we'll be talking about the comparison of 5 and 20 weeks. That is the time at 5 weeks when they're not protected with 20 weeks when they are protected. Okay, so let's go back and take a look at the particular events that we're interested in. So the initial events that I've told you about are the establishment of a beachhead, the small infected founder population, and its subsequent local expansion. And let's start with the cervix. And a particular portion of the cervix. So as we go from the ectocervix, a multi-layered squamous cell barrier here, into the transformation zone, into the single columnar epithelium that lines the endocervix. This transformation zone is certainly not the only site where you can get infection, but it is a site of greater vulnerability. And it's where we usually see these infected founder populations, both because it's a site of vulnerability and because you can examine rather exhaustively the tissues in that region. So if you do that in a naive animal and you use in situ hybridization to operationally define productively infected cells, and we've actually shown that it's true that an RNA positive cell is also a cell that's making virus. And we look at it in transmitted light. These black cells right here are viral RNA positive cells. The arrows point to interepithelial. They turn out to be CD4 T cells. They're in this endocervical region in the endocervic and transmission zone. This happens to be at day 4. The earliest we've seen these founder populations that we detect is around day 3. That doesn't mean there's nothing going on. It's just simply because there's a sampling issue when you're looking at tissues. And even if you do 25 or 50 sections, as you do, you may very well miss these founder populations. But certainly by day 3 and 4, we may see these founder populations. And when we look at the progression of infection at that local site, and we map those founder populations by representing them by these little green crosses, you can see this little cluster. This is on a two-dimensional grid or map. You can see these little green clusters here at day 4. This is day 7, and what you see is not some diffuse spread of infection, but you actually see the enlargement or the growth of that founder population. And out here at 10 days, you can see these massive coalescent viral RNA positive cells, and they're actually located in this transition zone. It's where the arrow points here. This is the ectocervix, this is the endocervix. These are founder populations that have expanded here in the endocervix and at the ectocervix. And this little line here actually represents a sort of serpentine path where the virus is, the infection is actually going along an inflammatory response. So the beauty from the virocentric perspective here is it elicits this immediate response from the immune system, and that counterintuitively acts against the host because it brings all these CD4 T cells into that region, and so the virus, it infects those cells and actually goes along this serpentine track, these darker staining cells. So what happens with vaccination? So this was a challenge at 20 weeks. On the left, I'm showing you in the green these SIV RNA positive cells at 4 days and then at 10 days, just to give you a feeling again for what they look like. They're green because instead of in transmitters, if you use reflected light, this is the appearance of the signal, which are actually silver grains. What happened in the vaccinated animals is either we couldn't find anything in 4 or 5 animals, or we found 3 or 4 cells, or I think 8 cells here, I think at day 11. So there was a striking decrease in the detection of these founder populations, and even more impressively in this expansion. If you look now at tissue viral loads done by quantitative PCR, here was what I call in vivo virus growth curves. You see all of the naive controls get infected, and by day 7 to 10, we're up in excess of 10 to the 6th copy equivalents per microgram of tissue. All of the colored symbols here represent the vaccinated animals that are below the limits of detection. So when we got these results, we were struck obviously by 2 things. Was first of all the where. It's at that site of vulnerability at this junction between the ectocervix, vagina, and endocervix. But also the when, the earliest stages of infection, when we couldn't see anything in terms of the founder population and no expansion. Well, the rapidity of that effect and the fact that we actually didn't see any virus-specific CTLs at that site in significant numbers led us to suspect that this might be antibody-mediated. So we did a very simple thing. We extracted the proteins from the female reproductive tract and ran a Western blot, and that's shown here. What we saw is some high molecular weight bands staining at GP80 and 160 particularly 160. And what you can see is the intensity. Each of these lanes represents an individual animal. If you compare 5 and 20 weeks, you can see that there's an increase in the cervical IgGs reacting with what turns out to be an oligomeric form of GP41. So again, looking at the 5-week and comparing it with the 20-week when they're not protected or they are protected, this was the significant 5-fold increase that we saw in those antibodies. Now, those antibodies actually not known to me at the time, but certainly after I read up on them, these were described more than 20 years ago. They happen to be antibodies reacting, IgG antibodies reacting with an oligomeric form in the Western blots of GP41, actually trimeric GP41. They were seen many, many years ago. They're typical of acute HIV infections, acute HIV-2 infections, and acute SIV infections. So this turns out to be the major acute response in primate and human lentivirus infections. Now, we don't think it's oligomeric GP41. We think what these correspond to, what the immune system is actually seeing, are trimeric stumps of GP41. So as you may know, the density or representation on the surface of the lentivirus envelope, or the envelope, they're not very densely represented, probably around 12 or so of them. And the non-covalent linkage between GP41 and GP120 is quite loose, actually. So it's very frequent that these GP120 molecules fall off. They're shed, basically. And what they leave behind are these trimeric stumps in a post-fusion complex of what we'll refer to from now on as trimeric GP41. They're very immunogenic, and I'll come back to it, but these little black lines, you probably actually can't see it, but they're an immunodominant disulfide bridge loop for cluster 1 immunodominant epitopes. It's just a terrific immunogen. So these are the antibodies that we were seeing there. And what I'm going to tell you now about is the localization of those antibodies that led to the following model, where local antibody production at that site, as well as antibodies coming in from the circulation, are concentrated by the neonatal receptor expression in a specialized cervical reserve epithelium. And what that effectively does is concentrate antibodies right on the path of virus entry, and we think that concentration is the reason that these basically non-neutralizing antibodies are so effective. So we got onto that when a former graduate student of mine, Ming Zhang, stained the tissues for IgG. I was wondering whether or not these antibodies might be localized in the epithelium. And what we saw was, shown here in red, are plasma cells staining in red. You can find them at 5 weeks, just beneath the epithelium. But there was this striking increase, about a 2.5-fold increase in the IgG positive cells at that site. What was even more striking is this lining of red staining IgG positive cells just beneath the epithelium, the lining epithelium. And that turned out to be staining of the cervical reserve epithelium. Well, I didn't know very much about the cervical reserve epithelium. It was fortunate that you can Google and get the world's literature and read it in about 30 minutes. Not much is known about it. Pathologists have known about it for many, many a year. They're called reserve epithelium because in a woman's reproductive life, they're thought to guard against pathogens, both as an anatomical barrier, and what we're now seeing is that they're actually a beautifully evolved immunological barrier as well. So the cervical reserve epithelium is a subcolumnar epithelium that is closely opposed to the lining epithelium. So staining, and I'll walk you very briefly through this slide, but basically the brown staining cells here are reserve epithelium marked with antibodies to cytokeratin 5. And this confocal micrograph shows the plasma cells in green. And in the merged image with cytokeratin 5, you can see that the reserve epithelium, cytokeratin 5 positive reserve epithelium, is also positive for IgG. Now, with somewhat characteristic immodesty, these images reminded me of a paper I had seen in PNAS by the Zhu laboratory in murine uterine epithelium, staining for the neonatal Fc receptor. So we stained for the neonatal Fc receptor, and that's shown here. And you can see, and shown here, you can see they basically co-localize, and that's what these confocal micrographs show us. So what we've visualized then is actually a generic system. Let's start with that. It's a generic system for concentrating IgG. And just to remind you all, in the female reproductive tract, the IgG is the major story, not IgA. It's probably, there's about 100 to 200-fold more IgG in the female reproductive tract. So it's clearly a system that's meant to concentrate and deliver IgG, and there was about a 9-fold increase in the staining for IgG. Now, of course, we're specifically interested in GP41 trimeric antibodies. And so James Voss at the Scripps Research Institute in Dennis Burton's laboratory created this soluble reagent to use in what's called reverse immunohistochemistry. Reverse immunohistochemistry because instead of using antibodies to go looking for antigens in cells. In this case, you're using a reagent of the antigen, basically, to go looking for antibodies within a cell. So this area here, above the what's called the membrane proximal external region, all the way around to the fusion peptide, is the fragment, a soluble reagent that James created for us that lacks the hydrophobic cytoplasmic and memper regions and the fusion peptide as well. And after playing around a lot with the conditions, we got some very beautiful staining with that, and I'll show you that on the next slide. So now we're using reverse immunohistochemistry with this reagent to stain in brown cells that contain GP41 trimeric antibodies. And what you see here at lower magnification and then at increasingly higher magnification are the staining of the cervical reserve epithelium that are chock full of these antibodies to trimeric GP41. Here's the lumen, here's the lining epithelium, here are the brown cells that represent the cervical reserve epithelium. There are also positive plasma cells here, and there's also concentration of the antibodies in these small blood vessels. So what we have here actually is a beautiful system both to deliver, actively deliver these antibodies into the tissues. We have a mechanism to produce those antibodies, and we have a mechanism to concentrate them at the mucosal front line. And so when we now compare these panels on the left and the right, these are the 5-week animals. You see very faint staining. You see the very intense staining on the right and 4 of the 20-week animals, showing again that the correlate of the maturation of protection is with these, the concentration of these antibodies. Now the neonatal FC receptor, just to remind you, was originally described by Bramble in terms of homeostasis, which is its major role in the adult. It's a saturable receptor. It moves IgG bidirectionally. And what we think we are seeing here is because of the local concentration and production of antibodies, this neonatal FC receptor layer is actually concentrating those antibodies precisely where they're needed. So that's the system that we've seen by staining the tissues, looking at the tissues, looking at the organization of this system in terms of female reproductive tract immunology. So that's the when and the where. Now I talk a little bit about the how, and I can do that very briefly because we do not actually understand how these antibodies work. The panels on the left, without going into details, show neutralization activity, and there is a maturation of neutralizing activity between 5 and 20 weeks against Tier 1 or tissue culture lab adapted viruses. But this lower panel shows that there's really no distinction between 5 and 20 weeks, at least in a test tube, and this peculiar neutralization curves that you see there. This is in the presence of complement. So it isn't that these antibodies to GP41 don't have any neutralizing activity, but in a test tube, it's not particularly impressive, and it certainly leaves behind a very large non-neutralizing fraction. But the mass action concept here is the way this system is constructed. It would basically concentrate the reactants, antibodies and virus, to give mass action effects that I don't think you can actually predict from these test tube experiments I'm showing on the left. And I don't know if Tom is up yet, but Tom Hope has described, and I think rightfully so, that antibodies in the mucus and at the surface can form basically a binding surface that will bind and trap virus and prevent it from reaching the underlying target cells. And just as a aside, these antibodies to trimeric GP41 are just the absolute champions in terms of binding antibodies. Well, that's the story for the transformation zone in the endocervix. If we now go, because we know that transmission can certainly occur vaginally as well, if we now look at what's going on in the vagina in these vaccinated monkeys, we see something really quite similar, but it's also, again, beautifully anatomically appropriate. So if we just look again at the extracted fluids, in the lower panels are vaginal IgG comparing 5 and 20 weeks when they're not protected or protected. You see exactly the same thing as we saw in the cervix. You see about a 5-fold increase in these antibodies reacting with GP41. And we see similar effects in terms of the virus growth curves, probably because the founder populations as they exist in the vagina are so spread out, we just don't routinely sample them. But when we simply look at the tissue viral loads, there's the same about six order of magnitude reduction in the vaginal tissue loads. Now when we look at the system in the vagina, it has some interesting and comparable features. The same magenta cells here, the plasma cells, the increase at 5 weeks that you see in that panel in two ways here. One, the other thing that we saw in the vagina, and we actually see at all mucosal sites with vaccination, are the induction of what are called ectopic or tertiary lymphoid follicles. They have some considerable structure which resembles the secondary lymphoid organs. I'm not going to go through that data today. But they also contain plasma cells that are staining brown here. They represent, again, by reverse immunohistochemistry, the plasma cells that are making IgG. And we have the same increase between 5 and 20 weeks in those follicles. What's really interesting about the vaginal surface, and what we're increasingly referring to as a mucosal immune system axis, is that there's a very active role that the epithelium plays in the recruitment and the delivery of antibodies. The green staining here is for CXCL10. It's also true for CXCL9. These are attractants for plasma cells. So you have green staining here. The yellow staining I'm showing here are plasma cells, both in the beneath the epithelium and in the ectopic follicles that are IgG positive. And it turns out that the neonatal FC receptor is mainly concentrated in this very basal and peribasal level of the epithelium in the vagina. So what that effectively does is it takes IgG that's being produced beneath that epithelium and it concentrates it in this basal layer. And so that's shown over here. Here's the lumen, and here's this red staining area that you can see with the concentration of IgG. It's even more dramatic post-challenge. Here's day 0 comparing a naive animal with day 4. The more plasma cells here, obviously before challenge, but within 4 or 5 days, there's this tremendous increase in plasma cells, IgG positive plasma cells, just beneath that neonatal FC receptor border. And if you do a kind of the perioscope scan of that, what you can see is this intense magenta staining for IgG. That intense staining actually represents two things. It represents the IgG positive producing cells that are located here, but it also represents the concentration of that IgG in just this basal layer. So we call it an IgG wall because it puts together plasma cells with the neonatal FC receptor in this axis. This is now staining again specifically for those IgG trimeric GP41 antibody producing cells. Here are plasma cells here. Day 0, here's a tremendous increase even by 4 days. Same thing in the ectopic follicles. These are Ki-67 positive plasmablasts within 4 or 5 days that are producing these antibodies. So just to sum up for the vaginal side of things, what we have is, again, a system including ectopic follicles and plasma cells to produce IgG and specific IgG. We have the neonatal FC receptor located at the basal layer. So if you're thinking teleologically and in terms of evolution, you have this multi-layered physical barrier in the ectocervix and vagina. but a particle that had penetrated that barrier would confront the maximum activity and concentration of IgG antibodies at the interface between that mucosal barrier and the target cells in the underlying submucosa. So that's what I have to tell you about the principle of timing and location and the local concentration and dependence. I'm going to spend a few minutes on a second principle, and that has to do with the local expansion and the recruitment of target cells. So it has to do with inhibiting the innate response that paradoxically recruits and facilitates transmission by bringing in CD4 T cells for fueling local expansion. And that's a tale of a second Fc receptor, the Fc gamma R2b receptor, which is the only Fc receptor with inhibitory activity. So what we're going to talk now about is a tale of two Fc receptors. The second one is this, the Fc gamma 2b receptor. And I'm going to talk about it first in terms of what it does to the recruitment of target cells, but it does it in a way that I think is beautifully designed again for the immune system to have a way of distinguishing between a pathogen it has never seen before and one that it's already seen. And that system works as follows. So we've seen that there's a system here for producing and concentrating antibodies at this mucosal surface. So imagine what happens now in the challenge. You have a high concentration of antibody as one of your reactants. You have a high concentration, at least initially, of virus. And what happens is you form immune complexes. Those immune complexes, it turns out, can then interact with this green staining layer. This is the lining epithelium now that has the inhibitory receptor. What happens is that elicits a very interesting genetic program in the epithelium of the expression of inhibitory molecules that inhibit NF-kappa B pathways and the various chemokine recruitment pathways. And what that does then, it blocks the recruitment of new target cells and thereby inhibits the local expansion. So let me just run through the background on that. For the local expansion, we have this founder population. The boundary conditions, I'll simply remind all of you, is not favorable for the virus. There are dispersed target cells, sometimes in aggregates in the vagina, but generally scattered lymphocytes. So the way the virus gets around that is it elicits this innate immune response immediately, and it brings in target cells. And the way that it does that is that interactions with the mucosal epithelium and the virus inoculum leads to the expression of MIP3 alpha, or CCL20. That brings in CCR6 positive plasma cells as well as macrophages. They elaborate beta chemokines. The beta chemokines then, in their turn, bring in the new target cells for expansion. And this happens really very quickly. So the brown staining cells are CD4 T cells in uninfected animals. Here's 1 day, not much change. By 4 days, you can see an influx of brown staining cells, but by 6 or 7 days, there's just this massive influx of target cells. So what that does is it changes the landscape immediately for very favorable conditions for virus infection. This just shows the target cells that have accumulated. And first of all, at lower magnification, this is a montage of zillions and zillions of sections that Leon Schein created. But the white staining cells here are CD4 T cells. The red staining cells here within this circle are the viral RNA positive cells. There's a little higher magnification. Again, you can see here's the lumen up here. Here are the CD4 T cells. And in those clusters where they're maximally concentrated is where you see these viral RNA positive red cells. So the virus really utilizes this system for expansion. Now, I've shown you the effect of comparing naive and animals, and vaccinated animals at 20 weeks. Here I'm showing you the comparison of the accumulation of target cells at day 4 in a naive animal versus what actually struck us immediately about the vaccinated animals. They look as though they've never seen virus at all. They're absolutely quiescent. And the reason for that is the T-cell recruitment is blocked. There's no expression of MIP3 alpha. The PDCs don't come in, and as a consequence, there's no local expansion. So what was the way we got at the underlying mechanism is by comparing microarrays before and after infection in naive animals with before and after infection in the vaccinated animals. And it turned out there's an incredibly small overlap of 1%. So they're very distinctive microarray things. But the important thing for this talk is that there were moderators of inflammation that would increase close to 4-fold in the vaccinated animals. And the arrows are pointing to some molecules that we did a rapid learning curve on, spread1, COMD1, and ROR-A. These turn out to be localized to the lining epithelium. So what was clear is that we had the epithelial expression of the inhibitors of NF-kappa B and recruitment in the vaccinated animals. And so eventually we got onto the idea of immune complex signaling in the unvaccinated animals, leading to inflammation, the innate immune response in the delta F vaccinated animals, and inhibitors of inflammation. And by localizing the Fc gamma 2b receptor, we then had basically a circuitry for the interaction with complexes with an inhibitory receptor, shutting down these chemokine and inflammatory pathways. And I'm simply showing that here. Here in green is the expression of the gamma R2b receptor. In the confocal micrographs, what we find is the co-localization of the inhibitory receptor, the antibodies to SIV, the antigen to SIV, all merging in this superficial lining epithelium. And because of time, I'm not going to go through the explant experiments, but basically what we did is in cervical explant cultures show that we could recreate this with immune complexes, and we could block it with antibodies to the gamma 2b receptor. So now to summarize what we've seen about the correlates of protection, there are two correlates of protection with the delta F vaccine. One is the production and concentration of antibodies. Correlate 2 is an inhibitory pathway to inhibit the recruitment. I want to take a little time just to finish up with what I call the practical side of this. So on the fundamental side, what we've seen here is that vaccination reveals this immune system axis with the epithelium, literally the front line, and thus new territory to explore, which we're certainly doing. But on the practical side, what it tells us is that if we can recreate this system of producing and concentrating antibodies, it might have protective effects. And more importantly, if we could do it with immunogens that don't have the safety issues of SIV delta F vaccine, that would be great. So that's what we've been doing for the past 2 or 2+ years. And the immunogens that we've designed basically start with the reagent that Voss created for us, this reagent right here, this soluble form of trimeric GP41. With Matt McCauley, we've displayed it at very, very high density on nanoparticle liposomes. And it turns out that you can incorporate the TLR4 agonist, MPLA, in these same particles. So we have an immunogen that has turned out to be just wonderful. We've also created for intranasal immunization, an FC form of that that we think will bind and may be effective in getting antibodies into the female reproductive tract. This was based, again, from work on the Zhu laboratory described a number of years ago, but basically incorporates this FC mutant in eliciting antibodies for the female reproductive tract. Here's the liposomes, here's the sham. And of course, with my overactive visual imagination, I see this little fuzz on the surface here. We're doing some immunogold so we can convince you. But anyway, there's a very dense array of the GP41 trimers on this surface. Along with the CpG9 TLR9 agonist, these immunogens have been just great. Here's with the last boost, and we have comparable antibodies against the trimer in 4 monkeys, which are comparable or exceed the levels that we find at 20 weeks in the vaccinated animals. So, and in the CBL. So the last couple of slides simply show, first of all, on the left are what I've shown you with SIV Delta Nef vaccine. These are the immunogens which in Western blot give us comparable, if not actually greater levels than with Delta Nef. And by reverse immunohistochemistry in these animals, here again is the neonatal FC receptor staining cervical epithelium, staining now for antibodies specifically for GP41 trimer. So this should be an interesting year. We've gone all the way down this pathway on the active immunization through necropsy and tissue analysis that I've just shown you that recreates what I call the tissue criterion of Delta Nef protective correlate. And down here we have, is it going to work? So we are in a year where we are going to be doing probably in about 6 months protection experiments against this very high dose challenge. And hopefully in the future I will have news. Maybe good, maybe not. But as I've often said, it's what I call the inconvenience of science, scientific data at times. However it comes out, it will be what you want to do in science for all of you. You want to do decisive experiments to see whether you're right or wrong. And in this case, maybe we could be right. So let me end by thanking the people in my laboratory, particularly Qingsen Li, who initiated these experiments, and Ming Zhang. Jeff Lifson and Mike Piatek have done the tissue viral loads. The monkey experiment, the pilot was done at Yerkes with Francis Villinger and Ken Rogers. Great kudos, and thanks to James Voss and Matt McCauley at the Scripps Research Institute. And all of these experiments over the years have been done with Paul Johnson, who is now director of the Yerkes Laboratory. And with that, I will conclude. I'd be glad to take questions.